First of all, I'm sorry for those of you who heard me talk last year. Excuse me, because a lot of what I'm going to talk about is what I talked about last year. And I think it's very important for us to go over all of this over and over and over again until we really understand. My first point, and this is a very biased view, this is my view, so please feel free to disagree with me. Please feel free to raise your hand and ask questions at any time. Now I'm going to start off by saying biotechnology, plant breeding, means nothing to feeding 9 billion people unless we can transfer that technology and the plant breeding to the farmers. Unless we can communicate with the farmers and tell them why they're going to need to grow what we give them, we're not going to make much progress. Secondly, I look at biotechnology in a very diverse manner. I look at biotechnology as from transgenic plants to a new tractor. New tractor's not bio, but it is technology. And for the farmer to produce biology and crops, he might need a new tractor. So keep biotechnology very broad-based. If we look at what's going to happen in the world over the next 30 to 40 years, the graph on the left is FAO's projection for wheat production between 2000 and 2040. So even though funding agencies and governments and universities and administrators are telling us we do not need any more wheat production, you can see that to feed 9 billion people, we're going to need to increase wheat production. And FAO projects that it will be increased from right now, we're about 2.4 billion tons to a little over 3 billion tons in the next 20 to 30 years. The graph on the right at the top is animals, meat production. The world is going to demand more meat production. So the increased wheat, increased corn, increased forages has got to be taken care of to feed animals to provide us more meat. The interesting two are the two graphs on the bottom. The one on the left tells us not the land that is under production, but the amount of land under production. The amount of land under agriculture production has not changed since World War II. Roughly 660 million hectares of land in the world is under production. It's obviously not the same land, but we've more than doubled yields on the same amount of land. That's due to farmers, plant breeders, biotechnologists. The one on the right is grazing land. So the amount of land under pasture for animals is projected not to change. Okay, how is plant breeding doing? Let's take biotechnology and go back. Get everything out of my pocket. So technology is, I'm going to talk mainly about plant breeding, because whether it's transgenics, gene cloning, gene sequencing, you all have to go through the plant breeder to the farmer. So how is plant breeding doing? Good in, and since I made this slide, I changed many to some crops. Slow or static or poor in other crops. Wheat is a problem crop because wheat is a polyploid. Barley, rice are much easier to do biotechnology, much easier to do transgenics in than wheat, because wheat's composed of not one species, but 3 species. So you've got one species within wheat buffering the other. To digress a bit, all organisms from humans to rice to wheat have about 30,000, 35,000 functional genes. So if each of the 3 species that comprise wheat have 30,000 functional genes, that means that some copies in wheat can be turned off and others turned on. So wheat is a very complex organism to work on from a molecular point of view. However, progress is slow, but progress is being made. Do we need biotechnology? Sometimes yes, sometimes no. For example, a lot can be done in wheat without the use of molecular biology, without the use of transgenics. We just came from a research program in England at the University of Nottingham, where they're crossing wild species of grass with wheat, and they're putting genes from not only other species, but other genera back into wheat. Now, I'm going to digress a bit and define a GMO. A GMO defined by the United Nations is any organism where you put one gene from one species into another, bypassing the sexual process. In other words, no sex. The people in England are using emasculation and pollination, so they're transferring genes from one species into wheat using the sexual process. So they're not GMOs. So when people talk to you about GMOs, ask them, how do you define a GMO? What do you mean? Because I can guarantee you everything but land-raised varieties of wheat in Tunisia have a gene from another species in them. Genetically engineered, not genetically modified. We need them. We need biotechnology, and there's many aspects in all crops, and we'll talk about some specific examples in wheat. Only if it's quicker. Because when you talk to Ravi Singh, where did more go? When you talk to Ravi Singh and the people from Simon later on this week, they select hundreds of millions of plants per year in the field. The best selection tool you'll ever have are your eyes. To do molecular markers, you need high throughput. Very, very high throughput. 4 weeks ago, we were in Monsanto in St. Louis. They can cut a small piece out of a seed, still have that seed viable to plant, and plant it, and they can cut out a small piece of the seed, analyze the DNA, and they can do 5 plants, 5 seeds per second. So that's high throughput, but that's a very expensive high throughput that no university, very few companies can afford. So it's got to be quicker. It's got to be more accurate. It's got to be more efficient. And for you folks as breeders and administrators, it's got to be cheaper. Every program, no matter whether it's my program or your program or Monsanto's program, we're all limited by funding. We need to look at new crops. We need to look at new approaches to bringing genes from other species into wheat, rice, olives, tobacco, any crop. Historical achievements of biotechnology, and I use this in the broadest sense, in wheat, strawberries, any crop, legumes, bringing in genes from other species. A new biotechnology impact that works very well is double haploid production, especially in barley. Micropropagation, which means tissue culture. And a technique that is not being used much anymore, but is highly appropriate, is mutation technology. If you go to the Atomic Energy Agency website, and look for the number of varieties, there's probably about 4,000 varieties of agronomic plants around the world that are the result of mutation breeding. And mutation breeding does not qualify, will never qualify as a GMO. Why? You're not bringing in a new gene, whether it's without sex or with sex, you're modifying an existing gene. So it's a much different technology. How do we define biotechnology? And what do I define it as? I think the best approach we can use in my wheat program would be marker-assisted selection. Excuse me. Gene discovery, this is what they're doing in England and other places. They're finding new genes in other species and putting them back into wheat. Genomics, that's studying the whole plant system at the molecular level. ESTs are expressed sequences, large libraries, microarrays, and gene cloning. There's a scientist at the University of California, Davis, that's cloned the leaf rust resistance genes. And we'll talk later about how you can pyramid them up using molecular markers in your wheat programs. Tissue culture, gene transfer from unrelated species. Genetic engineering. And please feel free, I will leave this presentation on this computer. So all of you students, ask Joseph to have the presentation so you can get all these words later and write them down, if you want. What do we need? What do I need in wheat? New sources of genes. For example, abiotic stress, aluminum tolerance is a very disastrous problem in many parts of the world. The most tolerant wheat in the world is tolerant to about 32 parts per million aluminum. If the soils become more acid, wheat production will go down drastically. We have sources from rye that are tolerant to over 200 parts per million aluminum. So if we can get these new sources of genes and put them back into wheat, we could do a lot of wheat improvement for growth on acid soil. Not only do we have to find those new genes, but we have to find a way to access these genes. We have to find a way to select these genes, both lab-based and field-based. And it has to be cost-effective. Everybody has a cost limitation to their program. Marker-assisted selection is a good technology. It's working well, but it's not new. We've been selecting markers and using them to select good genotypes since about 1990. The reason it's been slow in coming on is we needed high throughput. You've got to be able to analyze several thousand or tens of thousands of plants a week. You can't just analyze 100, 200, 300 plants. It's been around since the 80s, mainly used in breeding programs from the 90s. But now it's more effective because we've got high throughput. I'm getting ahead of my slides, sorry. What we need and what we're accessing now is more available markers and marker systems. And you folks that are taking classes well know that the number of marker systems, the new markers are coming out almost, it seems like, on a weekly basis. However, for us and even the companies, we need to get more cost effective. Here's a list of target markers for wheat at the present time. One of the problems with a lot of these markers is, especially like the abiotic stress. If somebody wants to do markers for aluminum tolerance, I'd say, why? All you need to do is grow your segregating populations in the field under acid soil. This plant survives, this plant dies, selection's easy, you don't need markers. However, if you want to pyramid up and put 2, 3, 4, 5 genes in one plant, to select for all 5 of those genes at the same time in the field is very, very difficult. If you select for 5 markers on one PCR reaction, then you can select for all 5 genes at the same time. When the people from CIMMYT get here, talk to them about their rust in the CIMMYT program, stem rust especially. Ravi Singh and Pawan, they're all trying to pyramid up multiple copies of different genes so that you don't have a single gene causing resistance. Why do you not want a single gene causing your resistance? The reason I, the reason I do not want a single gene causing resistance is if you have a single gene, you put a massive amount of selection pressure on the pathogen to overcome that single gene. And in those cases, my money, if I were going to bet, I would always bet on the pathogen. Okay? So if you have 3 or 4 genes and you have multiple levels of resistance, the selection pressure on the pathogen is a lot less. So that theoretically, the variety will last a lot longer. So this is a small list in wheat. If we had corn, if we had olives, if we had barley, we'd have a different list. What are our challenges? We need better and more diagnostic markers in most crops. And not can we, I'm positive we will make marker-assisted selection even more cost-effective. The challenge I feel today is for QTLs, which are quantitative traits. So instead of having a single gene for resistance, you've got a region of a genome, a quantitative region. So there could be 30, 40, 300, 400 genes in this region. So to find a marker to select for 300 or 400 genes in a region is much more difficult than to find a marker to select for 1 gene. Because lots of value-added traits in wheat and in other crops are governed not by a single gene, but by multiple genes. Multiple genes, quantitative traits. What are our challenges? More and accurate diagnostic markers, much higher throughput. In my program, I could be happy with doing 10,000 plants a week. Ravi Singh and CIMMYT, he would like to 10 times that, 20 times that, right? Ravi would like to do 1 million plants a week. Lower cost, no matter what your program, no matter who's funding you, we're always going to be limited by cost. The perfect markers. What's a perfect marker? A perfect marker is you actually take the gene sequence itself. Say the gene for RHT, which is the dwarfing genes in wheat, or the VRN, which are the vernalization genes, and you sequence and get that gene. Then you make a molecular marker from inside that gene. So when you analyze a PCR-based reaction, you're analyzing for that gene. Not the region near the gene, you're analyzing for that gene, which means it's a perfect marker. Conclusions for marker-assisted selection. It's making an impact, throughput's increasing. It will be and continue to be a major tool in plant breeding. But we need more targets, and we need not only more targets, we need better data analysis of how to manipulate marker-assisted selection into the plant breeding tool that you use, that Lud and I used in the field with our eyes. So, better analysis and coordination of the material, especially a move to select for complex traits. The major challenges, there's a massive amount of information out there. You students are generating more information on a weekly basis than we did in a year. How do we analyze that? How do we assemble it? How do we make sense out of it? Not only for publications, but how do we make sense out of it in the field as plant breeders? Do we see the benefits of genomic research? Sure. But we need more effective model to crop transitions. And what's a model? A model would be Arabidopsis. Everybody in this room knows what Arabidopsis is. It's a tiny little weed. But it's got a very small genome. It still has 35,000 genes. And the generation turnaround time is very rapid. You can make 100 generations a year, plus or minus. So you could do the genetics in Arabidopsis and transfer that information to wheat. Not only in plants, but in animals. For example, my son works in a cat genomics lab. Cats, as in four-legged cats. And he's looking at breeding populations for the genetics, studying the genetics of feline leukemia virus. Why feline leukemia virus? That's the closest virus in the world to AIDS virus. So they can study feline leukemia virus in the genetics of cats and transfer it, to potentially transfer it to humans. Same thing. You could study the genetics of height in Arabidopsis, and maybe that gene is similar to the height gene in wheat. So when you think about what you're doing and what I'm talking about, all of these things could be changed to animals, let alone plants. How have we done? In August 2014, in Science magazine, they published an article about enhancing yields and GMOs. This is in the United States, and this shows the yield on wheat through 2000 through 2020. The green line, this is in the United States. The green line is new and improved wheat varieties that have come out of companies and come out of modern breeding programs. So the yield increase in wheat, as Amor would say, I see him now, as Amor would say, wheat yields around the world seem to be plateauing. We were increasing at about 1% plus a year. Now it seems to be 1%, slightly less than 1%, and there are a lot of people that say wheat yields are plateauing. I don't believe that. This shows that wheat yields are not plateauing at all. If you look at GMO, this is the only slide I'm going to have of GMOs. This is corn in the United States. GMO corn. Now what has happened is that about 90+% of the corn acreage in the United States is GMO. First of all, there's been no scientifically documented evidence that eating corn grown in the United States is going to cause you problems because it's GMO. What GMO corn and soybeans in the United States have done, they've significantly decreased production costs on the farm. You don't need to put a tractor through the fields as many times as before. You don't need to use as many insecticides. The herbicide usage has drastically gone down, as you'll look at this. This is insecticides, but if I put up a slide with herbicides, you'd see the same thing. Okay, so in the United States, in terms of production, in terms of damage to the environment due to the use of chemicals, due to the use of gasoline to run your tractors, tires, et cetera, has significantly decreased. We can argue about this over coffee. The barriers to going from Arabidopsis to wheat or cats to humans is conceptual. stop, think about what you want to do in your plant, whether it's humans or wheat, and say, okay, how can I best study this problem? Can I use Arabidopsis? Can I use rice? Can I use barley? Or can I use cats and humans? Think about the fundamental science, whether it's plant breeding or cat breeding. How do we bridge the gap? It's easy. You folks are going to bridge the gap. Amor and I are too old. We're not going to bridge any gap. But you're going to be able to bridge the gap very quickly. The biggest problem we all have, whether it's animals or plants, is funding. 5 minutes? Okay. You're going to read this, so I'm going to go through because I want to give you an example of technology. See what happens when you talk too much? I want to give you an example. Many years, well, it seems like many years ago, it was in the late 90s, 2000s. I was consulting for the Atomic Energy Agency, and they have a plant breeding section. And I encourage all of you to look at that plant breeding section and try and get funding for your projects from Vienna. Joseph knows how to do that, he says. So we went into the Altiplano in Peru. The Altiplano in Peru, this is Lima. You go up over 5,000 meters down into the Altiplano, which is about 4,000 meters. That goes from Ecuador, Peru, Bolivia, down into Argentina. The reason we went there is that when we started this project, you could buy wheat in England, ship it to Lima, and the freight was $19 a ton. To go from Lima 300 kilometers into the Altiplano was $98 a ton. So basically the Peruvian government said if the Quechua Indians and the people that were living in the Altiplano wanted to survive, they had to produce their own food. This is why it costs $98 a ton to move grain into the Altiplano. This is what the Altiplano looks like, still looks like today. A very broad area, good for agriculture. And what we found up there was an old barley variety that the Spanish brought in the 1500s. It's like this tall, late, lodged. So we decided to use the existing landrace barley variety because it was something that the Indians and the people that were living there could see. So they were familiar with the morphology of this plant. So we decided to see if we could shorten the plant up, get better yield, and minimum of biotechnologies. So we used haploids, which is very simple technology. They had a cesium source in Lima, so we could irradiate the seed. We could regenerate it. We made double haploids because there were no PhD plant breeders in Peru at that time. So we wanted, if somebody made a selection, every generation they grew that selection, it would be identical. We went up, rented land in the Altiplano, grew everything the way the people that lived there grew it. These are the double haploids in clumps. And you can see some are early, some are late, some are short, some are tall. Every one of those clumps was a different plant, and they could be selected for very quickly. We were very lucky. We produced a plant that was 100 centimeters tall instead of 2 meters tall. 1 meter tall instead of 2 meters tall. It was 2 weeks earlier in maturing. It was morphologically identical to the Spanish landrace variety. And it was a naked kernel. That meant it was good for human food, not only for animals or deer. So right now, in the Altiplano, in Peru, instead of 2,000 hectares of the old landrace barley, they're a little over 300,000 hectares of this new cultivar, growing from 4,200 meters down to 3,500 meters in elevation. So when you look at your own projects and you look at transferring technology from the lab to the field, think about the region that you breed for. Civet is totally different. They're breeding for the world. I was breeding for the Altiplano in Peru. Okay? You might be breeding for all of Tunisia. You might be breeding for one small region of Tunisia. So think about the crop you're working on and think about the region that you're working in. And sit and look and say, do I need to do PCR reactions? Do I need to gene clone? Or can I do it much easier? There's nothing wrong with technology, only if you need it. I'd like to thank the 3 people that supplied me all the slides and that I've talked to for many years: Norman Borlaug, Peter Raven, and John Snape. Thank you very much for listening.